Hey everybody and welcome to Breaking Biotech. Today I'm going to talk about the AnaptysBio press release on their peanut allergy data, and I'm also going to talk a little bit about my biotech portfolio. So I have it listed up right here. It includes all the companies that I've shared with everyone except for a couple, and maybe I'll do specific posts on that, just on why I think they're interesting and why I hold them. So this is it. I'm going to update it weekly and see how the profit and loss is looking. I bought a lot of these in 2017, so it's not fair to compare year to date, but I've also got up here the year to date for the S&P 500 as well as XBI, the fund that tracks all of the biotech companies, so we can compare how I'm doing to the whole market and to just the biotech fund itself. And I'm also going to track volatility a little bit. I think that it's useful to do that. So as this goes, we'll see how volatility compares week to week. So Madrigal I've gone through, Aimune, Adamas, and that's it. So the other ones that I have in here are Gilead, Illumina, Fate, and Spark. So I have different reasons for holding these guys, and I think it's a topic for another show. I think Illumina, they have a lot of sequencing technology that they're pretty much the gold standard now, so I think they have a lot of potential. Spark is involved in gene therapy, and they have interesting candidates to help with blindness. Fate, I think, is a potential buyout opportunity, and I'll talk about that, why, maybe in another post. And Gilead, I think, is just a juggernaut on the scene that is involved in some good things. So anyway, overall, the profit and loss, Illumina's really been saving me here because it's been a bit of a disaster. So overall, I'm up 1.4% since I bought everything. The total biotech index is up 0.6%. So I guess year to date, I'm not doing that bad. But the real ones that have been holding me back are Adamas, and that's based on all this uncertainty regarding whether or not their competitor is going to be able to take in a lot of their market share. So we'll see about that, but a lot of these for me are long-term holds. So Madrigal and Adamas I'm going to be holding for a long time. Aimune as well actually is going to be a pretty long-term hold too. So not really expecting to see short-term gains here, but it would be nice to see how it goes. So otherwise, so for volatility, the XBI, we're expecting almost 4% volatility this week, so a move of about 3 points, either upwards or downwards. The entire market environment right now is still very uncertain with the tariffs coming down and the interest rate hikes going on. It looks like there's going to be a lot less money available to go into the market as there was in the last 10 years. So that's going to be a new challenge for everybody moving forward, and we'll see how it goes. Anyway, so with that, I'm going to update this weekly and see how we compare to other things. So I'm going to get into AnaptysBio now. And the reason why I want to talk about Anaptys is because I focus on peanut allergy regarding Aimune, and I've also done a post regarding DBVT, which is another company involved in trying to desensitize patients. So AnaptysBio is a San Diego-based company, and they're valued at almost $2.5 billion. So they're still kind of a small company, but they're similar to Prothena, which I talked about last week, in that they generate antibodies that target specific proteins. And these guys seem to focus on inflammation, so they try to target antibodies that can inhibit proteins involved in inflammation cascades. And inflammation's involved in tons of different diseases, from what they're saying Alzheimer's now, in certain cases, to diabetes, to even peanut allergy and immune responses like that. So they have a specific technology where they take advantage of somatic hypermutation, which is a natural thing that happens, and they use this and generate antibodies in vitro. So they also happen to have a collaboration with Celgene in different ways. So much like Prothena, they generate these antibodies, and they can only take them so far down the pipeline, and they hope that other companies that have a lot more resources can take them further. So it seems like in the the model that they have is they want to get the drugs as far as they can and show as much data as they can without spending as much money to cripple them in case they have a negative trial. And we're going to see that as we get through this a little bit, how it seems like they're cutting corners to get data in a higher phase trial when maybe it shouldn't be at that trial level. And I don't know. So we'll see. So anyway, one of their drug candidates is AnaptysBio, which inhibits interleukin 33. So let's see. So interleukin 33, not to get too intense on the science behind it, but basically in different allergic responses, whether it's dermatitis or peanut allergy or asthma, there's some sort of input, and whether that's an irritant or the allergen or the environment, it leads to a cascade of signals going downstream to different cells and different molecules, and it is also, how do I want to say, it's logarithmic. So a single peanut allergen can cause a cascade of events where the eventual effect is many-fold larger than the amount of initial input. So interleukin 33 gets released based on some sort of stimulus, and this molecule leads to a large cascade of events, including mast cell degranulation, which is very, very important in things like allergic reactions and asthma attacks. So if you get mast cell degranulation, that can be to such a magnitude where you get these anaphylactic responses, or these responses where you can't breathe, and it's hard to come back from that. So if you can prevent mast cell degranulation, then you're going to stop the process at that level, and you'll lead to beneficial outcomes when people have these negative reactions to different foods or to the environment in terms of dermatitis or asthma. So this is the step that you want to stop, in my opinion, is the mast cell degranulation. And Anaptys has shown some in vitro data that using the AnaptysBio, they can stop a lot of mast cell degranulation due to IL-33 inhibition. So the compound inhibits IL-33, and then you get less mast cell degranulation in the presence of an allergen. That's the point. So they've shown good data with the dermatitis trials that they've done, but they now want to see if this can be used in peanut allergy for moderate to severe patients in peanut allergy. And we talked about this similarly in the Immune trial 2-3 weeks ago when they showed their data. And so we'll get into it right now. Okay, so in this trial, it was a 2A trial, so they wanted to do a proof of concept trial, so not a giant one like we saw with Immune. So it was only 20 patients, and here's the layout. So they did a baseline oral food challenge, which we remember from the Immune trial. So they give cumulatively increasing doses of peanut protein, and then once symptoms start to appear, they stop and they say that is what the peanut tolerance is before they see symptoms. So I think they really dropped the ball here because that wasn't abundantly clear in how they assessed patients at baseline, and I think that leads to the real weakness in this study, but we'll talk about that. So at day -14 is when they did this baseline food challenge, and then at day 1 they gave the single dose of AnaptysBio, 300 milligram dose IV. Then at day 14 after that, they did another oral food challenge and evaluated whether or not there was an improvement in the amount of protein or symptoms that the patients dealt with when they were given increasing doses of peanut protein. Okay, so this is the chart that has everybody confused. So they only had 20 patients in the trial, and it's hard to get any meaningful powered data from a sample size that is this small, in my opinion. But they took these adult patients, so you can see the average age is 30, so that's one difference from the Immune trial. Even though they did have an adult population, most of the people were children. So the average cumulative peanut tolerance at baseline was around 230 milligrams. So what this could be interpreted as is that at 230 milligrams, they started to see symptoms, so the allergist went ahead and stopped the stopped giving more and said that that's what your tolerance is. But it seems like what they did instead is they gave them all of this peanut protein, and some patients still had mild reactions at this dose. So instead of continuing to increase the dose, they just stopped the trial and excluded these patients from the study. And the ones that they included were the ones that were having moderate to severe reactions at 236 milligrams. So, and in the conference call, they kept bringing up how they follow these practical guidelines and that they're very standard, and this is this is the gold standard of peanut allergy evaluation. But it's not quite as black and white as that, as they'd like to say, and I think this is where the weakness comes in. So, this article here, I'll leave it in the comments, is where they come up with these practical consensus guidelines on peanut allergy testing. So we'll talk about that a little bit later. But just for some context, so the 230 milligrams of peanut tolerance compared here is about an order of magnitude higher tolerance than the patients had for the entry baseline food challenge in the Palisade trial for Amune. So they were not able to tolerate more than 30 milligrams before the allergist had to stop the trial and say that they had, mild symptoms. So in the Amune trial, they're very clear. And if you look at this statement, they said the dose is successfully consumed with no more than mild symptoms. So once the symptoms get from mild to moderate is when they stop the trial, and that is what the dose is that you can tolerate there. So this, I don't think, is what they did necessarily in this trial. So I don't know. There's some confusion there, but I'll get into it a little more. So once they did the baseline at day 1, they injected the ANB020, and then at the exit food challenge, or this is interim analysis, so they're going to be doing another analysis in about 20 days, I think. Actually, it's right here. So at day 45, they're going to do another food challenge, and just to see if this effect maintains or not. So the data that they show here is that of the patients that received ANB020, about half of them had improved peanut tolerance to cumulative 500 milligrams at day 14 in the oral food challenge, and they had improved symptom severity at day 14 as well, 38% of them. So this is where it gets a little confusing. So they looked at symptom severity as well, and they kept the max protein, peanut protein, at 500 milligrams, which is also not ideal. If some patients could have gone to higher peanut protein, and then you could have evaluated whether or not there was actually a difference there. So the placebo only had an N of 3 because they excluded 2 patients that were apparently mild in severity, and that's really the distinguishing factor that they use. So they looked at if you are mild, you were decided to be a mild patient if you didn't show severe symptoms, not whether or not you could tolerate a more peanut protein than the moderate to severe patients. So you can either characterize patients in how severe their symptoms are, or what their maximum dose of peanut protein they can consume without getting moderate to severe reactions. So it seems like these lines were blurred in this trial. So in this 46% group, they were able to reach improved tolerance, but they don't tell you what the tolerance was. And then they also showed that at whatever tolerance that was, they had improved symptom severity. So I don't like this, because if you just pushed all the patients until they got to moderate to severe symptoms, then we could actually find out what the maximum cumulative dose that they could take before they get a severe reaction. And that's really, I think, what the goal is, because it's a tangible thing that we can do. The mild, moderate, severe symptoms, they're real, but they're more ethereal, and they're not really like a concrete thing that we can write down. So I think if they had have done more of a concrete thing, like this is the peanut protein at which that we had to stop the trial because they would have had a moderate reaction, it would have been better instead of this. So they do have a number at which point they were able to improve the tolerance, but they don't tell us, and they said that they're going to save it for a paper or an abstract at a conference. So we'll see what it is. But they also did show the improvements in severity of symptoms. But you could argue that, did they just give them less peanut protein to get those symptoms? We don't really know, because we don't know what their actual amount of peanut protein they were given. And if we look at, the maximum amount of peanut protein they gave was 500 milligrams. And we see from the baseline that they were able to tolerate 230 milligrams before getting really severe reactions. So I don't know. To double the amount of tolerance is nice, but if they're, I don't know. So this is why it's sort of confusing. And if you compare it to Amune, they're able to tolerate 600 milligrams compared to 30 when they start. So this is an order of magnitude increase in tolerance, whereas here they're only getting a twofold improvement. And now we don't know if the improvement could be better if they were able to increase the cumulative dose past 500. So I appreciate that this is a proof of concept study, and they're just trying to see whether there's any effect from their drug from one single dose of ANB020, but I almost think this leaves more questions unanswered than it does questions answered. And hopefully in their future study, they're going to have more patients, and they're going to be able to answer those questions better and be more clear in their guidelines and what they're looking for. Some good things, though, is that they didn't have any adverse reactions, which is something that you did see with the desensitization therapy. And I think that just comes to the way that they're attacking the problem is characteristically different. Providing people with the allergen is going to give them a lot of symptoms, and that's just comes with the nature of the game there. But in this case, you're not expecting to see very many adverse events because you're not providing the peanut protein itself. So it'll be nice to see whether or not they can come up with a dosing regimen to get sustained improvement and a more tangible improvement in improvement in peanut tolerance. And in the question and answer period, they were asked this repeatedly, but a lot of the answers that they gave I thought were kind of cop-outs. So, because they were asked whether or not, like, what's up with this high baseline compared to other studies like the Amune and the DBVT trials. And he talked about how this was an adult group, and adults are more likely to have a higher tolerance of peanut protein, which, it's arguable. But he also talked about how they didn't want to exclude any patients because it's not a representation of the real world in terms of people who might have a high tolerance, but once they hit that tolerance, they get very severe reactions. So I don't know. You could argue that if you have a lower tolerance of peanut protein, that should consider you to be a moderate to severe responder to peanuts, and they didn't make that very clear in terms of their presentation to me, anyway. Okay. Other than that, so in their future study, they're gonna hopefully fix these issues and see what they can get. So they did talk about that in their Q&A, and they went back to saying that this is just a proof of concept trial, and this is what it is. So they're expecting to get results from their where is it here? So in their eosinophilic asthma trial, they're doing another 2A, and when I was earlier in the video talking about how they're trying to just get as much data as they can to show other companies who are potential buyout players that, hey, we did this little study and we showed this. Is it attractive enough for other larger companies to come in and fund a big trial? So I think this is what they're trying to do in all this, and it for a single candidate to have all these different indications would be a huge benefit. But I don't know. I almost think that these guys spent a lot of time to do this peanut allergy study, when if they had done it more comprehensively, they could get a real answer on whether or not it does help with peanut allergy, which this still leaves a lot of questions. So he did say that they're going to tell us what the actual amount of peanut tolerance was in these patients at a upcoming conference, so I think we can look forward to that. I personally am not going to be investing in these guys yet. It's hard to tell whether or not this compound is good. I like the antibody strategies in general. I think are great, but I just don't know. Like if you're saying you're going to give people a twofold increase in improvement in their peanut tolerance, I don't know if that necessarily means that you're making patients get a tangible improvement in that way. And if they can already tolerate 230 milligrams of protein, that's almost a single peanut. And when it comes to what patients want is they want protection from anaphylaxis when they're going out to dinner or something like that. And if you're only selecting a patient population that can tolerate an entire peanut, I don't think that's what you're looking for. So I think they need to be more careful with that criteria when they get these baseline patients. So with that, guys, I'm going to wrap it up there. If you like what I'm doing, please like, subscribe, leave me a comment. And with that, I'll see you next time.